fellow viewers, welcome once again to the ongoing series on Mendelian genetics. If you remember, in the last 3 lectures, we had tried to justify the title Beyond Mendelian Inheritance, which means we were talking about deviations from Mendelism, where the gene interactions like incomplete dominance, codominance and multiple alleles, they were showing a behavior which was a little at variance with the normal expected Mendelian ratios of 3:1. Today's lecture focuses on another of the deviations, which is the pleiotropic genes. But before dwelling into that topic, let me just hurriedly complete a very important missing link which I had left on the last turn, and that is the multiple alleles in plants. Now, the multiple alleles in plants were for the first time observed by 2 scientists, East and Mangelsdorf. They discovered the genes for self-sterility in Nicotiana, that is tobacco, and they found out that fertilization was possible only when one of the alleles was absent, not otherwise. Now, this was a very interesting thing, because in terms of the plant being, say, self-pollinated or cross-pollinated, the basic idea was that the 2 parents should not have a similar genotype. If they were having a similar genotype, say for example S1, S2 and S1, S2 for the other parent, then this was a case of complete incompatibility. If one of the genes was different in a parent, then it led to a partial sterility or incompatibility. It was only when the genotypes of both the parents were different, then some sort of abortiveness was circumvented. So, that means this was also a very interesting idea about the multiple alleles. Viewers, you will be surprised to know that Oenothera, which is the evening primrose, possesses about 37 alleles which are responsible for self-sterility, and Trifolium, which is the red clover, has more than 200 such alleles. Recently, the DNA analysis of a Brassica S6 gene has also shown that it transcribes information for a specific glycoprotein, so which is recognized by an antibody. Now, this type of a reaction is something like an antigen antibody reaction, which we call in terms of embryology as the compatibility and incompatibility. So, that means definitely these different alleles would be having different DNA sequences. Here we are trying to display a chart which tells us as to what would be the functional policies of the plants if they are having multiple alleles. You would see in the first place that if both the parents are S1, S2, then it is absolutely 0 as far as the seed set is concerned. Let's then come to the main topic of the day, which is pleiotropic genes. As you can see on the screen, a gene which is going to exhibit multiple phenotypic expressions. Till now, we were talking about one gene, one character. Okay, one gene having many alleles, but the character was the same. Now, this is a different story altogether. This is a gene which is having multiple phenotypic expressions. Talking about the history, Pleitropy actually term has been derived from the Greek root, which means pleion or more, and tropos, which means way. So, that means there is more than one way that a particular character could manifest itself, or let us say in other words, more than one way in which a gene could function and manifest itself. So, that means when one gene is going to influence 2 or more seemingly unrelated phenotypic characters, I would stress the seemingly unrelated phenotypic traits are very important. We would justify them in our coming discussion. So, to define pleiotropy, it is basically the phenomenon where a single locus of a gene affects two or more distinct phenotypic traits. This term was actually introduced formally by a German scientist, a geneticist, Ludwig Pleit, in 1910, when he was writing an article in a Vorschrift, which means in honor of one of his teachers who had retired. You will be surprised to know that a keen observer like Mendel also did not miss out the idea of pleiotropy. It never escaped his keen eyes. And this monk described an early case of pleiotropy in his classical 1866 paper, as you know, which was written in German, and the English translation of the title was Experiments with Plant Hybridization. And he took those 7 characters, and the character number 3 was in fact a brown seed coat, violet flowers and axial spots. They seem to be somehow coming together, and likewise a white seed coat, white flowers and the lack of spots. They were making one more cluster. So, Mendel noticed that the flower colors were always correlated with these two other features. We have been talking about the law of segregation, the law of independent assortment, that the inheritance of one character is independent of the other, maybe in the next generation or in the gametes. Now, this is a different story altogether. It was found that the color of the seed coat and the color of the axils, they were somehow connected. And somehow the white flowers, wherever they were, their axils in the plants were colorless and the seed coats were also colorless. And where we had the purple flowers, they always had the reddish axils and brown gray seed coats. So, that means this was a character. For example, the flower color, this is what he thought, was actually affecting 3 characteristics and not just one. So, it was something like a 3-in-1 situation. So, he said that these 3 characters are found always together, and therefore, they must be under the control of a single factor. He did not go beyond that, but it was a very, very keen observation which took about 50 years to understand and to ramify later, and he did not make further elaborations. Today, what we can interpret Mendelian ideas in this regard would be that when we are talking about the flower color, it definitely specifies a protein which is going to cause the synthesis of a colored substance, say let us say a pigment, and this protein itself is going to work in different parts of the pea plant, be it the flowers. Why only flowers alone? In seed coat and in the leaf axils. So, that means these 3, say for example, the flower color, the seed coat and the axils seem to be absolutely unrelated. Their morphology is different, yet these seemingly unrelated, which I had stressed about 5 minutes back, the seemingly unrelated phenotypes can be traced back to perhaps a defect in one of the gene with several metabolic functions in different situations. So, that means if one of these pleiotropic genes goes defective, it would reflect a change in not just one character, but a series of seemingly unrelated characters, because the basic ingredient which was changed was also being used as a raw material for various metabolic pathways. So, that means when we are talking about a pleiotropic gene, we should appreciate that if we are talking of 3 characters, all the 3 manifestations may not be of the same intensity. So, that means these multiple effects would not be equally manifested. They could be primary, which we call as the major effects, because this is the most evident one in terms of its expression, or they could be secondary, which we call as the minor effects. Mind you, the minor effects may even be less evident. Nevertheless, they are important. Let us take a few examples from plants to begin with. In cotton, it has been found out that the gene which is responsible for the lint, in addition, it is also going to control many other characters like the color, the height, the size of the ball, the number of ovules that this plant is going to possess, and even the viability of seeds. So, that means any mutation in this gene is going to affect the main character as well as the seemingly unrelated 5 other phenotypic attributes. Likewise, in case of sorghum, the major effect seems to be control of internode length, which means either a plant would be tall or it would be a little dwarf, but then this is the major effect. This very gene is also going to have minor effects of the length of the panicle and even the size of the leaves. Mind you, these 3 characters are the ones which are going to have a terrible impact on the productivity of this crop itself. Another example we find is in case of the lupines or lupinous species. Here we have a different variety and different hues of beautiful colors of flowers, and this happens to be the major effect of the gene. Whereas, we find that in addition to the petals, there are anthocyanin pigmentation patterns even in the vegetative parts and also in the seed coat. And to top it up, there is another character, that is the plant height, that is also constituting the minor effects. Coming to the animal kingdom, Drosophila, we find that there is a single gene which is going to influence the size of wings of this fruit fly. In addition, character of the balancers, the position of the dorsal bristles, even the eye color and the shape of its spermathecae, and in addition, of course, the fertility and the longevity of this species. So, that means one gene and so many manifestations. Coming to the animals like the tiger, Panthera tigris, we find that their gene for coloration constitutes the major effect, and then there is a development of relation between the eyes and the brains also. You must have seen the white tigers in the zoo. So, this mutant white tiger would have a white coat color and cross eyes, which would not be seen in the original relative. So, that means mutation plays a very important role in the pleiotropic gene, and therefore, a mutation is not going to change or alter just one color, but simultaneously it is going to alter many unrelated traits simultaneously, and therefore, it is very true that the gene which is coding for a product may be required in different target mechanisms, and of course, it may have the same signaling function, but then the manifestations would be different. So, there are very many mechanisms which can contribute towards the origin of pleiotropy. It could be at different levels. One is, of course, gene pleiotropy. This will occur when a gene product is going to interact with multiple other proteins, and as a result many other reactions are going to be catalyzed, and therefore their manifestations are going to be different. The pleiotropy could all be classified as developmental pleiotropy. That means, in this case, the mutations, they have had multiple effects on the resulting single phenotype. There could be what is called as selectional pleiotropy. That is, this is going to occur when the phenotypes have many effects themselves, and more importantly on the fitness, that is depending upon the factors such as age and gender. Now, fitness is a term which is used in evolutionary biology and also in population genetics. We could even define it as the capability of an organism to pass on its gametes, that is, pass on its characters to the next generation. That means it is in terms of its sexual competence. And lastly, we have another category which is called as antagonistic Pleitropy. Now, this is going to deal with gene mutations which may have advantages in the early life of an individual. However, they will have disadvantages later in life. We can illustrate this idea also with the help of several examples. One of the examples which is very popular as far as pleiotropic genes is concerned is the frizzle gene in chickens. Now, we have different types of poultry. The frizzle gene actually is it's a case of mutation, and mutations in the gene for the feathers. Now, these feathers are going to be, unlike the normal ones, they are going to be curled outward and rather than being flat. So, as a result, what is going to happen? This is also going to affect, because it's a pleiotropic gene, this is also going to affect faster metabolism and enlarged organs of the individual. The functional point of view would be that the curling of the feathers is going to lead to loss of body heat, because there is less of insulation, and this would definitely require a faster basal metabolic rate in order to maintain homeostasis. So, you would find that such poultry would have many changes. They would consume more food, they would be infertile, and even if they are not infertile, their sexual maturation would delay. So, that means here again there is a pleiotropic gene which is controlling a major effect as well as various smaller or minor effects. No discussion is possible in non-Mendelian genetics without sickle cell anemia. Whether it is a case of incomplete dominance, codominance, pleiotropy, or even in our subsequent lectures of lethality, sickle cell anemia is always going to be there. And you remember from our earlier lectures that it was just a single mutation which was of the missense type, which led to the damage of so many multiple organs, including the heart, brain and lungs. And of course, the major issue was the sickling of cells. Another classical example of pleiotropy happens to be phenylketonuria. It is an autosomal recessive disorder, and there is a defect on the single gene which is located on chromosome number 12, which codes for the enzyme phenylalanine hydroxylase. This particular enzyme converts phenylalanine to tyrosine. See, we have in our dietary components, we require these amino acids, namely phenylalanine and tyrosine. And the phenylalanine so formed in our body has to be converted to tyrosine, because tyrosine is the starting material for melanin and for various other dopamine and other proteins. So, once this chain is broken, because the enzyme phenylalanine hydroxylase is non-functional, then the whole thing stops. As a result, there is going to be a vast accumulation of phenylalanine in the body, and this is going to damage the nervous system in case of infants, and this would result in so many disorders, including the mental faculties, seizures and heart problems and various other delays in development. If we were to look at the pedigree chart, and these are two carriers of the disease, and their marriage is going to have a probability of not 3:1, because one of them is going to die who has the homozygous recessive alleles, whereas the ratio now 3:1 gets modified to 1:2. There is another syndrome which we have, and by the way, if we wish to now call a syndrome as a dangerous pleiotropic manifestation of multiple alleles, it would not be wrong. Now, Marfan syndrome was also named after a French physician, Antoine Marfan, and this is basically an autosomal dominant disorder. The gene is present on the chromosome number 15. Q means that it is the larger component of the chromosome or the larger arm, and this encodes a protein fibrillin. Fibrillin is going to be responsible for the formation of connective tissue. So, if fibrillin is absent, then we will find that there is all abnormal connective tissue, whether it is of the heart, blood vessels, covering of the spinal cord, and various other internal organs like our eyes, bones and lungs. So, this is going to result in acute deformities. You can see on your screens how the spinal cord is curved and very long limbs, however less of coordination between them. Cystic fibrosis is another classical example, the gene of which is located on chromosome number 7, again the longer part or the longer arm. Now, this autosomal recessive disorder is also a life limiting one because it is chronic and progressive, and there is an acute respiratory inflammation, respiratory problems, and the insufficiency of pancreatic function. There is a frequent electrolyte loss resulting in even in male infertility. A very interesting aspect is that in this syndrome we find that one of the sequence gets deleted, and when one of the sequence gets deleted, then of course one triplet is less, the nucleotide is again changed. Talking of antagonistic pleiotropy, we again come back to our old well explained sickle cell anemia, because I told you in some of the earlier lectures also that why was a selection very important for the heterozygotes. They must be having some advantage which we had defined as the heterozygote advantage in populations who were present in the malaria infested areas. So, this is a theory which has been proposed to explain how the different aging process can be attributed to natural selection. As far as the pleiotropic alleles is concerned, we know that an allele is going to have a negative impact, but then it has to be sometimes favored by natural selection, because even if it has a little advantage, so there would be some goodness in its negativity also. As I said before, we can again exemplify this with the help of sickle cell trait, because we find that the pleiotropic gene of HBS, the mutation, it has a blend of advantages as well as disadvantages. The advantages I just mentioned, that and those who are homozygous for this HBS allele, obviously they will have a short life and there will be a damage to their multiple body symptoms, there will be a sickle cell trait and they would die. But then natural selection would favor those who are heterozygous, and they do not, even though they experience some degree of negative symptoms of sickle cell disease, yet the fact that they are resistant to malaria makes them possible. So, here is the frequency of HBS allele would in a population would always be higher in those regions where malaria happens to be very prevalent, even in an epidemic form. The ratio again boils down to 1:2 rather than the expected ratio of 3:1. Just to sum up the whole idea, it would be important to know that many more genes in human beings happen to be in fact pleiotropic. It has been found out that the genetic variation among the single nucleotide polymorphisms or the SNPs, which we generally call as the SNPs, mean that their one little change in the nucleotide is going to change in the whole scenario of the regulatory and the disease causing genes, and they would turn pleiotropic. So, more than 300 genes have been located in the human genome so far, which happens to be pleiotropic. Thank you.